Next chapter, reproduction in living things. Now in this part, we are going to look at asexual reproduction and also sexual reproduction in flowering plants. Reproduction is the process by which mature living organisms give rise to new organisms of their own kind. Reproduction is divided into two broad categories, sexual and asexual reproduction. Sexual reproduction involves the fusion of male and female gametes in a process called fertilization to form a zygote which develops into a fully grown individual. It occurs in animals and flowering plants or angiosperms. Asexual reproduction does not involve gametes, but rather a part of an organism develops into new individuals. It occurs in bacteria, protozoans, fungi, and some plants. Common modes of asexual reproduction include binary fission, budding, sporulation, and vegetative propagation. Binary fission in amoeba. This is the simplest form of asexual reproduction that involves the splitting or fission of the original cell into two similar daughter cells. Simple as it may sound, binary fission is a complex process involving internal reorganization of the cell nucleus and cytoplasm. When conditions are favorable, that is sufficient food and oxygen, and favorable temperature and pH, a mature amoeba divides. Initially, the mother cell elongates and the nucleus divides into two in a process called karyokinesis. The cytoplasm then divides into two through a process called cytokinesis, resulting into two daughter cells. This division is mitotic in nature. Each daughter cell has a diploid of chromosomes just like the parent amoeba cell. Budding in yeast. This is a form of division in which a new individual is produced as an outgrowth of the parent cell and develops into a separate independent organism. It occurs in yeast and hydra. When conditions are favorable, that is availability of sugar, and favorable temperature and pH, the yeast cell reproduces by budding. First, a small outgrowth appears on the body of the parent cell. As this grows, the nucleus divides into two daughter nuclei and one migrates into the outgrowth. Budding continues, forming a chain of cells. The buds detach by forming a constriction at the base. The daughter cells produced are generally smaller than the parent cell. A yeast cell has a nucleus, cytoplasm, vacuole, and a cell membrane. In hydra, little buds grow on the outside of the parent and then break off to form a separate individual. Next is spore formation in rhizopus. Rhizopus is a saprophytic fungi that grows on substrates like bread, rotting fruits, and other decaying organic matter. The vegetative body is called mycelium, which consists of many branched threads called hyphae. The horizontal hyphae are called stolons. The hyphae which grows vertically are called sporangiophore. The tips of these upright hyphae swell up to form sporangia, the spore-bearing structures. Inside the sporangium, the nuclei divide meiotically, producing daughter nuclei that contain a haploid number of chromosomes. The daughter nuclei develop into spores by the incorporation of cytoplasm around each nucleus and a wall around the cytoplasm. When fully mature, the sporangium wall bursts and releases the spores, which are dispersed to restart a fresh generation of growth and development. mushrooms, molds, ferns, and mosses reproduce by sporulation. The other uncommon forms of asexual reproduction include multiple fission, regeneration, fragmentation, and vegetative propagation. Multiple fission occurs during unfavorable conditions such as lack of water. A thick wall or cyst is formed around the amoeba. Then the cell splits many times within the cyst to form many amoebae. In regeneration, the body breaks into pieces and each piece can grow into a new individual. Starfish and planaria are capable of regeneration. In fragmentation, the plant body breaks into two or more fragments or pieces upon maturation. Each fragment then develops into a new organism. Spirogyra reproduces through fragmentation. Vegetative propagation is seen in some plants. Here, new plants are formed from the vegetative parts of other plants. Potatoes, for instance, develop underground tubers that can develop into new plants. Strawberries can propagate by runners, and so on and so on. And now we move to sexual reproduction in flowering plants. Now, the flower is the most conspicuous and the most attractive part of the plant. But we know that flowers are not present only to add beauty to the plant. They are the reproductive parts of the plant. A flower is a modified shoot consisting of a modified stem and modified leaves. A typical flower is made up of a stalk or pedicel, and the apex of the stalk is called the receptacle. On the receptacle are attached the four floral parts: the calyx, corolla, andrecium, and gynoecium. The outermost wall is the calyx, which consists of sepals that are green, leaf-like structures that protect the flower in its bud stage. The sepals can either be free or fused. When free, the calyx is called polysepalous, and when fused, the calyx is called gamosepalous. The next inner layer is the corolla, which consists of petals that are usually thin and soft. There is a wide variety in the color and shape of the corolla. It can be in the shape of a wheel, a funnel, or a bell, and sometimes tubular. The primary function of the corolla is to attract insects and birds for pollination. Like the calyx, the petals of the corolla can be fused or free. When fused, the corolla is called gamopetalous, and when free, it is called polypetalous. The positional arrangement of petals and sepals with respect to other parts of the flower is called estivation. There are several types of estivations. The common ones are velvet, twisted, imbricate, and vexillary estivations. In velvet estivation, the sepals or petals touch each other at the margin with no overlapping. In twisted estivation, one petal or sepal overlaps the next one, and so on and so on. In imbricate estivation, the margins of the sepals or petals overlap one another, but in no particular direction. And in vexillary estivation, each petal or sepal covers the petal or sepal that is within it. The third floral part is the androecium, which consists of stamens, which are the male reproductive organs of a flower. Each stamen has a stalk called a filament and an anther at the tip. The anther usually has two lobes, each with two chambers, which are the pollen sacs. The pollen sacs contain pollen grains. Stamens can also be free or polyandrous as in lily, or fused in varying degrees as in China rose, the pea plant, or the citrus plant. In China rose, the stamens are united in one bundle and are called monoadelphas. In the pea flower, the stamens are fused into two bundles and are called diadelphas. And in the lemon flower, the stamens are grouped into more than two bundles and are called polyadelphas. The last part is the gynoecium, or the pistil, which consists of one or more carpels, which are the female reproductive organs of a flower. The carpels can either be fused or free. For example, in tomato and mustard, the carpels are fused and are called syncarpas. While in rose and lotus, they are free and are called apocarpas. Each carpel has three parts: stigma, style, and ovary. The stigma is the part of the carpel that receives the pollen grains. The style is the elongated tube that connects the stigma and the ovary. The ovary is the enlarged basal part and bears one or more ovules which are attached to a placenta that is soft and flat. Once fertilization has taken place, the ovules form the seeds and the ovary develops into the fruit. The arrangement of ovules within an ovary is called placentation. There are different types of placentation. In axial placentation, the placenta is axial and the ovules are attached to it in a multilocular ovary, as in lemon and tomato. In parietal placentation, the ovary is initially single-chambered. A false septum later develops and divides it into two chambers. The ovules develop on the periphery or inner wall of the ovary. Parietal placentation is seen in plants such as cucumber and melons. When the placenta develops at the base of the ovary and a single ovule attached to it, the placentation is basal. This is seen in marigold and sunflower. In free or central placentation, septa are absent and the ovules develop on the central axis, as seen in primrose and dianthus. And in marginal placentation, along the central suture of the ovary, the placenta forms a ridge. The ovules are borne on this ridge in two rows, as seen in pea plant. Ovaries can also be classified based on their position with respect to the receptacle. An epigynous ovary is one whose position on the receptacle is such that all the other floral parts occur above it. An example is in an apple flower. A hypogynous ovary is placed on the receptacle and above all the other floral parts. An example is in hibiscus. When the receptacle surrounds the carpel, with the other floral parts arising around the ovary, it is called perigynous. Now, if a flower has all the four floral parts, that is the calyx, corolla, androecium and gynoecium, it is called a complete flower. If one or more of the parts is absent, then it is called an incomplete flower. calyx and corolla are called the non-essential parts, while gynoecium and androecium are called the essential parts of the flower. Most flowers contain both the male and female reproductive parts. Such flowers are called bisexual flowers, like pea and China rose. On the other hand, some flowers have either the male or female reproductive parts. Such flowers are called unisexual flowers. The male flower is called a staminate flower, while the female flower is called the pistillate flower, like in maize plant. For unisexual flowers, if both the male and female flowers are born on the same plant, such a plant is called a monoecious plant. An example is the maize plant. Notice that the male and female flowers are born separately, but on the same plant. However, there are cases where the plants bear either the male or female flowers. Such plants are called dioecious plants. Next, we move to pollination. Now, for sexual reproduction to take place, a male gamete is to be fused with a female gamete to form a zygote. For this to happen, the male gamete has to reach the female gamete. The pollen grain carrying the male gamete has to be transferred from the anther to the stigma of the same or different flower. This is called pollination. If the pollen grain of a flower lands on the stigma of the same flower, it is called self-pollination. But if the pollen grain of a flower lands on the stigma of a flower of the same or another plant, it is called cross-pollination. Pollination occurs with the help of different agents like wind, water, and animals or insects. Insect-pollinated flowers are large and conspicuous with scented petals and produce nectar to attract insects. The anthers are small and firmly attached to the filament to prevent breakage as the insects rub against them. The stigma are also small and sticky to allow pollen grains on the insect body to stick on them. The pollen grains are heavy and sticky in order to adhere to the insect body. Wind-pollinated flowers are less conspicuous, are not scented, and lack nectar. They have large anthers which are loosely attached to the flexible filaments to allow movements in order to release the pollen grains. The pollen grains are usually small, smooth, and light so that they are easily blown by wind. The stigma are long and feathery, hanging outside the flower in order to trap the flying pollen grains. Self-pollination limits the variety of progeny and may depress plant vigor. However, it can be advantageous, allowing the plants to reproduce in areas where pollinators' populations have been greatly reduced or are naturally unavailable. It also ensures there is preservation of quality genes in a plant species. Features and mechanisms that hinder self-pollination. Dioecious and monoecious plants. Dioecious plants have male and female reproductive parts located on different plants of the same species, like in pawpaw. This eliminates the probability of self-pollination. Monoecious plants, like the maize, have the male and female reproductive parts located separately on the same plant. This minimizes the chances of self-pollination. Protandry and protogeny. Protandry is a case where the stamens or the male reproductive parts mature earlier than the pistil or the female reproductive parts. The former releases pollen grains but cannot pollinate the pistil of the same flower. Protogeny is where the pistil matures earlier than the stamen, so they cannot be pollinated by the pollen grains from the anther of the same flower. Heterostyle is a situation of having different arrangements of the style and stigma. For instance, flowers could have shorter stamens than pistils and hence become impossible for the pollen to land on the stigma of the same flower. Self-sterility is a case where pollen grains cannot germinate on the stigma of the same plant, but only on a different plant of the same species. Double fertilization in angiosperms. Angiosperms of flowering plants undergo a unique reproduction process where there are two fertilization events. This double fertilization occurs between the male and female gametophytes. Before the fertilization events can occur, the ovules have to undergo some changes. Initially, the ovary contains one reproductive cell known as the megaspore or the mother cell. This cell is diploid and undergoes meiosis, producing 4 haploid megaspores. 3 of these megaspores degenerate, leaving only one surviving megaspore. The surviving megaspore expands and undergoes 3 rounds of mitosis to produce 8 haploid nuclei that share the same cytoplasm. This complete structure is known as the embryo sac. Within the embryo sac, cell walls begin to form around some of the nuclei. 3 cells, called antipodal cells, form opposite the opening of the ovule, known as the micropyle. Another 3 cells form just above the micropyle. 2 of these are synergids, and the other is the egg cell. This leaves 2 nuclei at the center of the ovule. These 2 nuclei remain together in one large cell. They are called the polar nuclei. It is the egg cell and the polar nuclei which will become part of the double fertilization event. What remains now is for the pollen grain to move from the anther to the stigma. The pollen grain contains 2 main cells. A tube cell makes the bulk of the pollen grain, and the sperm cell, which is at this stage known as the generative cell. This is a haploid cell. Notice that the generative cell is contained within the tube cell together with the tube nucleus. In the process of pollination, the pollen grain lands on the stigma. Once it lands, it starts to germinate. The tube cell forms a growing tube down the style, with the tube nucleus leading the way and the generative cell behind it. Once near the ovary, the generative cell divides by mitosis to produce 2 haploid sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the sperm cells fuses with the egg cell to form a diploid zygote, which will become the embryo. The other sperm cell moves on and fuses the 2 polar nuclei, forming a triploid cell, which will develop into an endosperm and serves as the embryo food supply during early development. Double fertilization only occur in angiosperms. The next thing after fertilization is fruit and seed development. The zygote formed during fertilization divides several times to form an embryo inside the ovule. The ovule develops a tough coat and changes into a seed. The ovary, on the other hand, grows and forms a fruit, enclosing the seed. The other floral parts fall off. And so, a seed present in a fruit contains an embryo, which is basically a baby plant. This baby plant can then grow into a seedling when provided with appropriate conditions for germination. Now, fruits. There are a number of ways in which fruits can be classified. One way is as true or false fruits. If a fruit develops directly from the ovary, such a fruit is called a true fruit. If some other floral parts participate in the formation of a fruit, such a fruit is known as a false fruit. A mango is a typical example of a true fruit. An apple is a good example of a false fruit. In an apple, the thalamus is modified to form a fruit. This is the internal structure of a true fruit of a mango. It consists of a fruit wall developed from the ovary and is called the pericarp. It is made up of 3 layers: epicarp, mesocarp, and endocarp. Mechanism of fruits and seed dispersal. Once fruits and seeds are produced, they need not germinate and grow in the same location. There are several agents that are involved in the transfer of seeds and fruits from one location to another: wind, water, animals, and even self-explosive mechanisms help in the dispersal of fruits and seeds. Physical features of the seeds and fruits determine the most effective agent of dispersal. When dispersed fruits and seeds have features like hair, wings, purples, and feathery styles. They also tend to be light in order to be carried over long distances. Water-dispersed fruits and seeds have floating features like waterproof fibrous outer coats. They also tend to be light in order to allow them to float. Some also trap air bubbles to increase buoyancy. Coconuts are dispersed this way. Animal dispersals occur in seeds and fruits with hooks, barbs, spines, or bristles by which they adhere to the animal's body. Some fruits may also be eaten up by animals, but the seeds are not digested. They are later released with the fecal waste somewhere else. Self-explosive is seen in legumes. The fruits have valves or jaculators. After the pod dries up, it breaks open and the seeds break from the placenta inside the pod and are thrown a distance away.